Good morning, everyone. We are still going to wait a couple more minutes so that everyone can connect and join us. Thank you very much for your patience. Good morning, everyone. We would like to begin by thanking you for attending this CondaLab talk, which is the second webinar of our food series. My name is Aletia Cruz, and I'm one of the microbiology product managers at Conda. We mentioned it last time, but for our first-timers, CondaLab Talks refers to the webinars we will be preparing for you from time to time on subjects of interest in different areas, such as food or water safety, to mention some industries. It is a pleasure to welcome you all, and the purpose of this CondaLab talk, as you know, is to discuss STEC, which is a real concern for health authorities around the world. I would like to mention that this platform has a question box, so please send us any questions or doubts you may have during the presentation, and we will answer these at the end of the webinar. So we will begin with the bacteria, the diseases this can cause, then we will review the European regulations in place, followed by a quick overview of STEC importance in the industry, and finally we will review the reference method. To start, we will introduce our beloved and well-known Escherichia coli, its serotypes, the diseases this can cause. So E. coli are typically gram-negative and rose-shaped bacteria with rounded ends. They are also non-spore forming and are usually motile through the action of periticus flagella, which refers to flagella projecting in all directions, such as in the image shown here. The bacteria can grow between 20 degrees Celsius and 40 degrees Celsius. They are facultatively anaerobic and produce gas from fermentation of carbohydrates, as seen by acid and gas production from lactose. E. coli is found almost exclusively in the digestive tract of warm-blooded animals and human beings as well, which is why its presence is used as an indicator of fecal contamination in food and water control. The grand majority of E. coli serogroups are non-pathogenic, but some can cause severe diarrheal disease. In fact, there are 7 main diarrheogenic pathobars of E. coli, which are a set of strains with the same or similar characteristics that are differentiated from other strains of the same species or even subspecies on the basis of distinctive pathogenicity. These are Shiga toxigenic E. coli, or STEC, which is also known as virotoxigenic E. coli, VTEC, which we will get into in a bit, or even enterohemorrhagic E. coli in older literature, referring to bloody stools that are often a part of the clinical presentation, and therefore sometimes this acronym, EHEC, continues to be present in some parts. The next one is enterotoxigenic E. coli, enteroinvasive E. coli, enteropathogenic E. coli, enteroaggregative E. coli, diffusively adherent E. coli, and a final pathotype, adherent invasive E. coli, which is associated with gastrointestinal diseases such as Crohn's disease. Each specific type causes diarrheal disease through different mechanisms, and each disease presents itself with different clinical symptoms. And as the name of our webinar implies, we will be focusing on the STEC pathobar. So STEC is unique among these E. coli by virtue of harboring and expressing the genes for Shiga toxins type 1, STX1, and 2, STX2, that are very similar to the toxin produced by Shigella dysentery. Worldwide, the terms Shiga toxin and virotoxin are interchangeable because they refer to their cytotoxic effect on the viro monkey kidney cell line. Therefore, STEC are often referred to as virotoxigenic E. coli, or VTEC. On the figure on the right, there's a phase contrast microscopy of the Shiga toxin effect on viro cell monolayers. In the first panel, A is a medium control at day 1, which are cells in continuous monolayer. Next, with STEC filtrate, we have image at B, day 1, C, day 2, and D, day 5, where we can observe the monolayer being destroyed. So when talking about STEC in the past decades, most attention has been focused on one particular STEC O antigen serotype, O157, O being the somatic antigen, and more specifically the one of flagellar antigen serotype H7, meaning STEC O157H7, which predominates in reported outbreaks and sporadic cases worldwide, although not universally. Transmission of these organisms is thought to occur mainly through contaminated foods. Nevertheless, there is an increasing awareness that several of the non-O157 STEC serogroups are emerging as important pathogens associated with sporadic cases of disease, as well as outbreaks of foodborne illnesses. Many non-157 STEC serogroups have been identified, but not all of them have been shown to cause illness. However, in 2011, a new strain of E. coli bacteria, O104H4, caused a serious outbreak of foodborne illness in Germany. From an initial case control study in May, the outbreak was associated with the consumption of fresh salad vegetables. Later, the O104 serotype was also incorrectly linked to cucumbers imported from Spain, since these did not contain the specific strain. Subsequent investigations showed that the risk of infection was significantly associated with the consumption of fresh sprouted seeds rather than with other fresh vegetables. A German organic farm, which produces a variety of sprouted foods, was identified as the likely source of E. coli outbreak, and an investigation of the production site showed no evidence of environmental contamination, and neither did the produce. Hence, contaminated seeds were for the sprout production were the most likely source. On June 2011, it was announced that seeds of organic fenugreek imported from Egypt were likely the source of the outbreak, and a month later the outbreak was declared over, with a total of 3,911 cases reported to the ECDC and WHO. Regarding non-0157 serogroups, in USA these are responsible for almost 60% of the cases every year. And also in a survey of non-0157 STEC outbreaks reported in the world, the most frequent cause of illness has been associated with serogroup 026, followed by 0111 and 0103. Surveys indicate that the non-0157 STEC strains make up a large portion of the STEC isolated from patients in developed countries, with the interesting exception of Japan, where 0157 is the major STEC serogroup. Let's move on to the diseases caused by STEC. In 2015, the WHO estimated that foodborne STEC caused more than 1 million illnesses. Therefore, STEC has emerged as a frequent cause of foodborne gastroenteritis, both sporadically and in outbreaks, creating a substantial risk of hemolytic uremic syndrome, HUS. STEC infections in humans were the third most commonly reported zoonosis in the EU and increased from 2014 to 2018. The EU notification rate had a 39% increase in 2018 compared to the previous year. The incubation period following ingestion of STEC is usually at the third day, but it can go between the first to 8th day, and duration of the illness is typically 1 to 12 days. The incubation period for illness is strain dependent, and this is largely due to the different pathogenic mechanisms exhibited. Symptoms consist of watery diarrhea, abdominal pain, fever, and vomiting. Also, hemorrhagic colitis, which is bloody diarrhea, is found in 90% of patients, and HUS is seen in 5 to 15% of patients. In the absence of HUS, the resolution of symptoms is spontaneous in most cases, which is a good news, because HUS is characterized by acute kidney failure, microangiopathic hemolytic anemia, which is damage of small blood cells, and decrease of platelets or thrombocytopenia. The kidneys are frequently attacked, but the central nervous system, lungs, pancreas, and heart may also be affected. A high proportion of patients are hospitalized. Some develop end-stage renal disease, or as we know it more commonly, kidney failure, which means that either a transplant is required or dialysis is obligatory. Death can also occur, unfortunately. Transmission of STEC to humans occurs through consumption of meat, foods eaten raw, such as lettuce or spinach, or unpasteurized milk or juices. Direct animal to human and human to human transmission have occurred. Water and soil that have been contaminated with ruminant feces can also be vehicles of STEC infection. Therefore, it is no surprise that contaminated irrigation waters can be a source of STEC in fruits, vegetables, and other produce, mainly because this serotype exhibits prolonged survival in water at low temperatures. It is possible that almost any raw food will contain STEC, and during meal preparation, cross-contamination may occur if careful hygiene is not practiced. To visualize the importance of STEC, we are looking at a chart from the European Union One Health 2018 Zoonosis Report, which was published last December. In 2018, STEC infections were the third most commonly reported zoonosis behind CAMP and Salmonella infections. It may not be as shocking as CAMP infections, however, it still accounted for 8,000 confirmed cases in the EU in 1 year alone. That being said, and taking into consideration what these infections can cause and all the possible contamination sources, it is no wonder it has become a concern for health authorities all over the world. We will review now some of the European Union regulations that are currently in place. It's starting with E. coli. We have Commission Regulation 1441/2007 on microbiological criteria for foodstuffs that has established a safety criteria for E. coli in different food categories as a fecal contamination indicator that we've seen before. These food categories include virtually every food, including those ready to eat, such as you've guessed it, sprouts. The analytical method of reference is ISO 16649 and all its parts. For the enumeration of beta-glucuronidase positive E. coli. It's a quite hard word to pronounce. This document establishes the sampling and preparation of samples and frequencies. Moving on, we have Commission Decision 2009-312 as regards dedicated surveillance networks for communicable diseases. It establishes a list of communicable diseases for epidemiological purposes. STEC infections are included in this list as a food and waterborne disease of environmental origin. For the communicable diseases listed, epidemiological surveillance within the community network is to be performed by the standardized collection and analysis of data. Next, we have Commission Recommendation 2012-66, where ISO 16654 is recommended for the detection of toxigenic E. coli. Also, a full validation of this method by the EU Reference Laboratory for E. coli has been performed and completed since 2014, including for milk and sprouts. Finally, we have Commission Regulation 209/2013 for the microbiological criteria that should be considered for these 6 serogroups that are recognized to be those causing the most hemolytic uremic syndrome, HUS cases, being 0104H4 the responsible, as we've seen, for the 2011 outbreak. Of course, it cannot be excluded that other STEC serogroups may be pathogenic to humans as well. It indicates that there must be absence in 25 grams of produce, and the reference method is ISO 13136, a PCR method for the detection of STEC and the determination of 5 specific serogroups. The sampling includes the irrigation water as well as the sprouts. And since STEC can be found virtually in every food, let's take a quick overview of its importance in the industry. In the EU One Health Zoonosis Report of 2018, we can find a chart that indicates attributed STEC foodborne disease burden to 6 food categories, plus the category other foods. In the European A subregion, beef was estimated to be the major food source, followed by dairy products and vegetables, as we can see. To reinforce this information, according to a 2018 WHO STEC report, the most frequently attributed sources of cases globally are fruits and vegetables with an attribution proportion of 13%, beef with 11%, and dairy products with 7%. So we have a coincidence in the 3 principal food sources, meaning 3 industries that have to take serious and specific actions for STEC. After reviewing this information, it is very clear the importance of controlling STEC for industries and health authorities. So now we will review the ISO method 16654 for the detection of E. coli 0157. This method is a 6-steps procedure, and all the products indicated and highlighted in bold are available at Conda. As always, if you'd like more information about our products, please visit our website www.condalab.com or contact us. To start, we must prepare an initial suspension. So to the sample, tryptic acid in soy broth modified with novobiocin, or MTSV, is added to obtain a ratio of 1:10, mass to volume or volume to volume. This is incubated at 41.5 degrees Celsius for 6 hours and subsequently for a further 12 to 18 hours. Then immunomagnetic separation, IMS, should be carried out after 6 hours and again, if necessary, after 12 to 18 hours of incubation. When you have the washed and resuspended magnetic particles, transfer these to a plate of McConkey agar with sorbitol, CT-SMAC, a first selective medium, and also to a second selective isolation medium for E. coli 0157, for which we recommend our Condachrome E. coli 0157. The incubation conditions for the CT-SMAC are at 37 degrees Celsius for 18 to 24 hours, and the same for Condachrome E. coli 0157, so you can comfortably incubate them together. On CT-SMAC, typical colonies are transparent and almost colorless, with a pale yellowish-brown appearance, and on Condachrome E. coli 0157, the colonies are pale pink. Next, take typical colonies from each media and spread onto a plate of nutrient agar. The incubation conditions are at 37 degrees Celsius for 18 to 24 hours. Afterwards, inoculate only one colony into a tube of tryptophan culture broth, incubate at 37 degrees Celsius for 24 hours. Then add 1 milliliter of COVAX reagent for the biochemical confirmation. And lastly, for the serological identification, you should only examine the indel-positive colonies, the ones with formation of a red color, for their serological reaction with antiserum to E. coli 0157. With this slide, the webinar ends. So now we will begin with the Q&A part. Let's see what questions we have. Okay, so the first question we have is, why do we recommend the chromogenic agar as the second medium? Okay, so we recommend Condachrome E. coli 0157 because, as I've mentioned before, the incubation conditions are the same, so it's very comfortable. But obviously, it's not the only reason. To start, it is a highly selective medium that inhibits any other bacteria. And second, when you use a chromogenic media, you are getting to know the result, even before confirming it with any serological identification or biochemical confirmation. So, for example, if you have a produce batch and you see pink colonies on the chromogenic agar, you have more time to react and make any recall decisions or any decision at all, which I believe is very valuable. Okay, so next, let's see what we have. Okay. Why is a modified McConkey used for 0157? Well, the strains of E. coli 0157 are kind of easy to isolate due to one of their unique biochemical characteristics, which is that this particular serotype is unable to ferment sorbitol. So in CT-SMAC, the lactose is replaced by sorbitol, and the red fennel being a pH indicator that when the pH in the medium decreases, the color will change from neutral red to pink. Let's imagine you have a plate inoculated. E. coli 0157 will then grow as colorless colonies since it doesn't ferment sorbitol, and other E. coli that does, which is the majority, will be pink. Also, and very important, is the cefixime telluride supplement that provides a highly selective recovery of E. coli 0157 H7 from other E. coli strains and other Enterobacteriaceae. And we have almost run out of time. Let's see. A final question, and we have a lucky one. Why is the IMS, immunomagnetic separation, indicated in the ISO method? So immunomagnetic separation uses specific antigen-antibody interactions, which are very similar to ELISA. However, rather than confirming bacterial isolates or their toxins, IMS is frequently used to concentrate the target pathogen directly from the enrichment media. It is a very straightforward method to concentrate pathogens, and it is widely used. So it works by utilizing a magnetic bead that is conjugated with target-specific antibodies. The magnetic beads are incubated with the initial suspension, as we've seen. Then a magnet is utilized to pull the bead antibody antigen complex out of the sample, which allows a very quick concentration of the pathogen of interest, while minimizing interference of background microflora. And that is the reason why it is utilized. You can have it under 1 hour, this method of concentration. So we have a couple more questions, and we will answer these via email because we've unfortunately run out of time. So thank you very much for attending our STEC webinar. We very hope to meet you in the next one. And if you have any further questions or any doubt, we'll answer these via email. And thank you very much for attending. Please reach us at micro@condalab.com. Have a great day, everyone, and please stay safe. Bye-bye.